Hey guys! Today we're going to talk about transport processes across the plasma membrane, a vital aspect of cell biology. The plasma membrane, also known as the cell membrane, acts as a barrier that separates the inside of the cell from the outside environment. But how do cells get essential nutrients, waste, and other substances in and out of the cell? That's where transport processes come in. Cells have a plasma membrane that regulates the movement of substances in and out of the cell. The membrane is composed mainly of phospholipid molecules and scattered protein molecules. The phospholipid molecules have a polar hydrophilic head and nonpolar hydrophobic tails, forming a bilayer with the heads facing out and the tails facing in. The inner part of the cell, called the cytosol, is separated from the surrounding interstitial fluid by the plasma membrane. Substances move through the membrane through either passive or active transport processes, with passive processes not requiring energy expenditure and active processes requiring energy. These processes are the transport processes that move substances across the cell membrane. We shall now delve into these transport processes, starting with passive transport processes. The passive transport processes include simple diffusion, facilitated diffusion, and osmosis. Diffusion is the movement of a substance from a high concentration to a low concentration with a tendency to spread evenly over a given space, such as a sugar cube dissolving in water and spreading evenly until reaching equilibrium. It involves the movement of a substance along a concentration gradient, where there's a difference in its concentration between two areas. So, molecules will move from an area of high concentration to an area of low concentration. In cells, this process is called cellular diffusion and occurs across the plasma membrane. Cellular diffusion depends on the concentration of the solute, a dissolved substance, in the interstitial fluid outside the cell and the cytosol inside the cell. It can happen through simple or facilitated diffusion. Simple diffusion happens with small, nonpolar solutes that can move through the nonpolar interior of the plasma membrane. To understand simple diffusion in an easy way, let's consider if the concentration of oxygen molecules outside the cell is higher than the concentration inside of the cell, the oxygen molecules can move into the cell through the plasma membrane by simple diffusion, down the concentration gradient, without any help. Conversely, if the concentration of carbon dioxide molecules inside the cell is higher, they can also cross the membrane by simple diffusion, exiting the cell and entering the interstitial fluid. Thus, simple diffusion involves nonpolar molecules passing straight through the membrane, without any help. Examples of materials that diffuse in this way include oxygen, carbon dioxide, and small fatty acids. Now, let's talk about facilitated diffusion. Facilitated diffusion is the second type of diffusion that occurs with the help of plasma membrane proteins for small, charged or polar solutes that are unable to pass through the nonpolar phospholipid bilayer by simple diffusion. In facilitated diffusion, polar or charged solutes that are too small to diffuse through the nonpolar phospholipid bilayer can enter and exit a cell with the aid of membrane proteins. Facilitated diffusion has two forms, channel-mediated and carrier-mediated. The distinction between the two lies in the type of transport protein used to move the solute across the membrane. Channel-mediated diffusion refers to the movement of ions, which are charged particles with a positive or negative charge, through protein channels in the plasma membrane. These channels are specific to one type of ion, that is, they allow only one type of ion to move through them, and can be either leak channels which are always open, or gated channels, which only open temporarily in response to a stimulus. On the other hand, facilitated diffusion using carriers involves the movement of polar, but small solutes like simple sugars, carbohydrates, or amino acids across the plasma membrane. This process is facilitated by carrier proteins that alter their shape to transport the solute. For example, glucose binds to a carrier protein, causing a change in shape that moves the glucose molecule to the opposite side of the membrane. Now, to the last type of passive transport process, we'll talk about osmosis. Osmosis refers to the passive movement of water through a selectively permeable membrane, driven by a difference in concentration of water on either side. In the cell, this can occur either by slipping between the phospholipid molecules that form the plasma membrane or through specialized protein channels called aquaporins.
The plasma membrane is largely impermeable to solutes like charged, polar, and large substances.
As a result, the concentration of solutes can vary between the cytosol and the interstitial fluid. For instance, if the interstitial fluid has a concentration of 20% solutes and 80% water, and the cytosol has 10% solutes and 90% water, water will move from the side with higher concentration, that is 90% in the cytosol, to the side with lower concentration, which is 80% in the interstitial fluid, to reach equilibrium,
when the concentration of the solutes on either side of the membrane is equal.
Now that we've gone through the passive transport processes, we shall take a look at the active transport processes.
Active processes in membrane transport require the expenditure of cellular energy.
These processes are classified into two types, active transport and vesicular transport. Active transport involves the movement of solutes against their concentration gradient, from areas of low concentration to areas of higher concentration. On the other hand, vesicular transport is the process of transporting large substances across the plasma membrane by means of a membrane-enclosed sac called a vesicle, which contains the materials.
Active transport includes two forms, primary active transport and secondary active transport.
Primary active transport utilizes cellular protein pumps known as ion pumps to move ions across the membrane against their concentration gradient. For example, when three sodium ions and ATP bind to a sodium-potassium ATPase pump, ATP breaks down into ADP and phosphorus, releasing energy that causes the pump to change shape and release the sodium ions into the interstitial fluid. Meanwhile, two potassium ions bind to the pump and the phosphorus is released into the cytosol of the cell. The pump then reverts back to its original shape, releasing the potassium ions into the cytosol. The pump becomes ready for the next cycle. Secondary active transport, on the other hand, involves the transfer of a solute against its concentration gradient using the energy derived from the movement of another substance down its concentration gradient. The solute that moves from an area of high concentration to low concentration provides the energy to transport the second substance from an area of low concentration to high concentration. That is, in secondary active transport, energy is derived from the movement of one substance down its concentration gradient to move another substance against its concentration gradient.
There are two forms of secondary active transport, symport and antiport.
In symport, both substances move in the same direction, using energy from the movement of one substance to transport another substance. For example, the energy from the movement of sodium ions from high concentration outside the cell to low concentration inside the cell can be used to move glucose from a low concentration area to a high concentration area in the same direction as the movement of the sodium ions. In antiport system, substances move in opposite directions, using energy from the movement of one substance to transport another substance. For example, the energy from moving sodium ions from high concentration outside the cell to low concentration inside the cell can be used to move hydrogen ions from low concentration inside the cell to high concentration outside the cell. Now to the other type of active transport process, we're going to talk about vesicular transport. In this type of transport, larger substances such as proteins or polysaccharides are moved across the plasma membrane. This process is divided into two types, exocytosis and endocytosis. In exocytosis, materials are expelled from the cell into the surrounding interstitial fluid. The Golgi apparatus produces membranous vesicles that transport these materials to the cell membrane, where the vesicle and plasma membrane fuse, releasing the vesicle's contents outside the cell.
The vesicle's membrane then becomes a part of the plasma membrane. In endocytosis, the plasma membrane folds inward to enclose a substance, forming a vesicle.
There are three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis.
Phagocytosis is a form of endocytosis where a large particle is engulfed by the plasma membrane folding inward and forming a vesicle around it.
The vesicle then fuses with a lysosome, which is a membrane-bound sac filled with digestive enzymes, to break down the particle into its constituent molecules.
Phagocytosis is often referred to as cell-eating.
In pinocytosis, the plasma membrane invaginates to capture droplets of the interstitial fluid that contains soluble substances, which the cell can utilize.
This process is also referred to as cell drinking. In receptor-mediated endocytosis, the plasma membrane uses special receptors to capture molecules from the interstitial fluid.
The receptors bind with the molecules and the membrane folds inward, creating a vesicle that transports the receptors and the bound molecules inside the cell.
It is important to mention that both endocytosis and exocytosis require energy for their completion, as they are active processes. So there you have it. These are the main transport processes across the plasma membrane.
From simple diffusion to complex active transport, these processes are essential for maintaining the balance of substances within cells and allowing them to function properly. Thanks for watching, and don't forget to like and subscribe for more educational content.